pattern, but they're not organized in a dense enough network that conduction is uniform.
Okay, thank you very much, Tim. Next up, we have Leia from National Heart Center, Singapore, talking to us about 3D scaffolds for endothelial cell regeneration from human-induced pluripotent stem cells. Thanks, Leia.
Good morning, everyone. I'm Lei Ye from National Heart Center, National Heart Research Institute, Singapore. Thanks, Jay, for the invitation. So today I will share you results using the iPSC for endothelial differentiation. So as we know, the endothelial cells are dynamic and play a key role in maintaining cardiovascular physiology. And although endothelial cells mediate signaling pathways that regulate myocardial vascularization, perfusion, metabolism, and also contractile performance. And the discovery of the iPSC is revolutionized the regenerative medicine and disease modeling. So previously we have discovered that in a 3D dimension, endothelium can be efficiently differentiated into endothelial cells. So based on this protocol, we further explored to develop a new one. So this is the old protocol. We use the actin A BMP4 to commit iPSC into mesodermal cells, and on day 1, then we switch to VEGF TGF with EPO continuously for 4 days, and on day 14, we will cell sorting to purify the cells. As you hear, the same 2 cell lines we use as a monolayer, and all 3D patch cultures we found actually in the monolayer using this same protocol, only about 7% or 5% cells differ into endothelial cells. However, and when these cells were seeded in 3D patch, we found they can increase to 31 and 45%. So based on this, this is how the iPS cell culture and the differentiation in the 3D model. This is showed how the iPS cell seeded into a fibrin patch. This is on the day cell was seeded as a single cells, and after 48 hours culture in 3D patch, they will grow from small cells, then into the small cluster of cells. Then after 24 hours, differentiating with actin A and the, then they will from small cluster into a cluster of the cells. Then after 4 days culture with VEGF, TGF, EPO, then they will from small cluster into a big cluster. Then this on day 11, they will, after release, they will form a monolayer. So basically, based on the old protocol, we use the 2 new factors to replace actin A and BMP4. And on day 11, we will sort the cells. So this modified protocols was tested in 5 cell lines. PCBC and the GIP was normal iPS cell lines. However, DP1, D indicates diabetes, and the progerin PG is indicates progerin. So we not only found these protocols effective in the normal iPS cell lines, they also very effective in the disease iPS cell lines. So biological duplicates, we found that actually more than 85% efficiency can be achieved across all 5 iPS cell lines. So these cell lines have a typical EC characterization. They can express CD31 and CD144 on cell surface, as shown here. They also can form tubular structures on matrix, as shown here. However, obviously, the disease iPS-derived endothelial cells they have poor tubular formation as compared to normal iPS cell lines. They also are able to uptake acetylated low-density lipoprotein, as shown here, red fluorescence. We also performed the flow assay to determine what's the phenotype of these cells. We standard them for DAL4 and epinephrine B4. We found the maximum about 60% co-express the epinephrine B4. However, more than 99% express DAL4. And also they do not express protoporphyrin on the cell surface, and also almost 95% pure for CXCR4. So we suggest that most of these cells generated in the 3D environment actually was arteriovenous endothelial cells. These cells have the capability to maintain their EC phenotype up to 4 weeks in cell culture. As shown here, they were more than 95% stand positive for CD31 and similarly for CD144. So biological duplicates indicate more than 95% actually co-express CD31, CD144 up to 4 weeks in cell culture systems. Then we also to determine whether these endothelials have biological function in vitro. So we developed a mouse model of ischemic limb disease. As shown here, 3 days before ligation, then we will perform laser scanning to visualize the perfusion in both legs. Then 3 days after ligation, we will again do the laser scanning to see the perfusion was totally blocked. So on the day, then we inject the either basal median or human endothelials. We found actually at 2 weeks after treatment, the animals did not receive endothelial transplant. They have totally limb loss. However, in the animal limb received the endothelial injections, they have the reconstruct of perfusion, which was almost 50% to the normalized after to the left limb. We found actually endothelial transplantation also induced the angiogenesis, and shown here the CD31. The ligated limb have recovered about 80% to left limb based on CD31 staining, indicating about new angiogenesis occurred in the limb received endothelial transplantation. However, the smooth muscle-based co-staining suggests the arterial density was almost similar between the ligated and also normal legs. At last, we also performing the immunostaining to determine the fate of transplanted endothelial cells. And shown here, most human endothelial cells, as shown here, stand positive for human CD31, was extensively survived in the limb, which indicated angiogenesis was achieved by the transplanted the endothelial cells. Also, some of the human endothelial was coated by mouse, small cells, indicating they also participated into the artery formation in the ischemic limb. We also performed some study to determine whether the human patient specific endothelials derived from the RPS have some dysfunction or not. Basically, we found that actually the disease endothelial cells, they have almost double the cell population doubling time as shown here compared with normal iPS-derived endothelial cells. They have lower G expression level as shown here by the ENOS quantitative RT-PCR assess. Also, we determined the NO release. We found actually both the PG and the diabetes cells, they have released less NO compared with normal, the endothelial cell level. So this data suggests we have developed a new and more efficient the endothelial definition protocols, which can achieve more than 90% efficiency in 3D scaffolds. And also the endothelial cell derived from this pro have biological function and in vitro and in vivo both. And also disease-specific endothelials have poor function as compared to normal iPS-derived endothelials in vitro. So this study was sponsored by the National Medical Research Council of Singapore and under my study, carried out by my staffs in my lab, and a collaboration with Stu Cook and Colin Stewart from IMB ASTA. Thank you for your attention.